Most people think ovarian aging happens because a woman gradually runs out of eggs. That is only part of the story. A major 2026 study suggests that the physical environment surrounding those eggs also changes. As the ovary ages, its once flexible tissue becomes increasingly packed with collagen. The organ grows stiffer, almost like soft soil slowly hardening around a seed. When researchers blocked one inflammatory signal in old animals, that tissue became softer. Hormone function improved, more follicles developed, and fertility increased. The signal was a protein called interleukin-11, or IL-11. The result does not mean scientists can reverse menopause or extend fertility in women today. Most of the treatment experiments were performed in mice and rats. But the research reveals a surprising possibility. Reproductive aging may be driven not only by the number and quality of remaining eggs, but also by the mechanical condition of the tissue in which they live. To understand why that matters, imagine an ovarian follicle as a tiny expanding sphere. Inside it is an immature egg surrounded by cells that nourish it, produce hormones, and help guide its development. As the follicle grows, it needs room to expand. Eventually, during ovulation, the surrounding tissue must remodel so the egg can be released. That process depends on the ovary being neither too soft nor too rigid. Some stiffness is useful. It helps keep dormant follicles inactive until the body is ready to recruit them. But growing follicles need a more flexible environment. If the surrounding matrix becomes excessively dense and stiff, expansion becomes difficult. It is like trying to inflate a balloon inside wet sand compared with hardened concrete. The researchers began by examining human ovarian tissue from different age groups. They analyzed samples from women aged 18 to 28, 35 to 42, and 47 to 52. They also studied tissue from people with premature ovarian insufficiency after chemotherapy, polycystic ovary syndrome, and ovarian endometriosis. Using atomic force microscopy, a technique capable of measuring the mechanical properties of extremely small areas, they found that ovarian tissue became progressively stiffer with age. The same pattern appeared in the disease groups. Ovaries affected by chemotherapy-related insufficiency, polycystic ovary syndrome, or endometriosis contained more collagen and showed greater stiffness than age-matched control tissue. This was important because it suggested stiffness was not merely a side effect appearing after ovarian function had already declined. It might be part of the process causing that decline. The researchers then placed follicles inside laboratory materials with different levels of stiffness. In a softer environment, follicles expanded more successfully. In the stiffer material, they remained smaller. The surrounding granulosa cells divided less and produced lower levels of estrogen and progesterone. The structure around a follicle was changing how the follicle behaved. But what was making aging ovaries harden? The team analyzed proteins in human ovarian tissue and studied gene activity in ovarian fibroblasts. Fibroblasts are maintenance cells that produce the extracellular matrix, the network of collagen and other materials that gives tissue its structure. When repair is controlled, fibroblasts are essential. When they remain overactive, they can produce too much matrix. Tissue becomes scarred and fibrotic. This happens in organs such as the liver, lungs, heart, and kidneys. The new study indicates that a similar process can occur in the ovary. One signal stood out. IL-11 increased with ovarian age in humans, mice, and rats. It was also elevated in ovarian tissue affected by premature ovarian insufficiency, polycystic ovary syndrome, and endometriosis. When human ovarian fibroblasts were exposed to IL-11 in the laboratory, they became activated and produced more collagen. The protein appeared to push these cells into a tissue-stiffening state. This created a potential cycle. Aging and inflammation increase IL-11. IL-11 activates fibroblasts. The fibroblasts deposit more collagen. The ovary becomes stiffer. Follicles then struggle to grow and produce hormones normally. Declining ovarian function may create further inflammation and tissue damage, reinforcing the process. The ovary is not simply getting older. Its internal landscape may be gradually turning against the follicles it is supposed to support. The researchers then asked whether interrupting this cycle could preserve function. They genetically removed the receptor used by IL-11 in mice. Without that receptor, cells could not respond normally to the signal. The ovaries of these animals accumulated less excessive matrix and remained less stiff. The mice showed improvements in ovarian function across several models, including natural aging, chemotherapy-induced ovarian damage, and a model resembling polycystic ovary syndrome. Then came the more clinically interesting experiment. Instead of permanently deleting a gene, the scientists used nanoparticles carrying small interfering RNA designed to reduce IL-11 production. They administered the treatment to aged mice and rats. The treated animals showed reduced ovarian stiffness and improved reproductive performance. More follicles developed, hormone-related measures improved, and fertility increased. That is the central result. The researchers did not create new eggs. They changed the environment around the eggs that remained. This distinction may become important for future fertility treatment. Most current approaches focus on stimulating the ovaries with hormones, retrieving available eggs, freezing eggs or embryos, or replacing hormones after ovarian function declines. These strategies can be valuable, but they do not directly repair the aging tissue surrounding the follicles. A therapy targeting fibrosis might address a different part of the problem. It could potentially make the ovary more supportive of the follicles it still contains. IL-11 is especially interesting because this is not the first time scientists have connected it to aging. In a 2024 Nature study, blocking IL-11 in older mice reduced several signs of age-related decline and extended median lifespan by roughly 1/4. Treated animals showed improvements in metabolism and frailty, although those findings remain preclinical and do not establish that blocking IL-11 will extend human life. Together, the studies suggest IL-11 may help connect inflammation, fibrosis, declining organ function, and aging across multiple tissues. But the ovary presents a particularly difficult target. Ovarian aging is not caused by stiffness alone. Women are born with a finite supply of immature eggs, and both the number and genetic quality of those eggs decline with age. Chromosomal errors become more common. Mitochondria function less effectively. DNA damage, oxidative stress, altered hormones, cellular senescence, and changes in blood supply all contribute. Softening an aged ovary would not automatically turn a 45-year-old egg into a 25-year-old egg. It may help remaining follicles develop, but it cannot erase every form of accumulated damage. There are also major safety questions. Fibrosis pathways exist for a reason. Collagen provides structure, and fibroblasts help repair injured tissue. Blocking IL-11 too strongly, in the wrong organ, or for too long could interfere with healing or create other unexpected effects. Researchers would need to determine the correct dose, timing, delivery system, and patient group. A treatment designed to preserve fertility before chemotherapy might require a different strategy from one intended for natural reproductive aging. A person with endometriosis or polycystic ovary syndrome may also have different underlying biology from someone approaching menopause normally. The human tissue data in the study were observational. They showed that older and diseased ovaries were stiffer and contained more IL-11, but they did not prove that blocking IL-11 in women would restore fertility. The fertility improvements came from animal experiments. That line must remain clear. Mice reproduce quickly, age differently from humans, and respond to ovarian interventions in ways that may not translate directly to patients. Many treatments that look promising in rodents fail when tested in people. There is currently no approved anti-IL-11 therapy for delaying ovarian aging, preserving natural fertility, or reversing menopause. No supplement or food has been shown to reproduce the effect. The study also should not be interpreted as a reason to delay family planning based on the expectation that science will soon extend fertility. Translating a discovery like this into a safe treatment can take years, and success is never guaranteed. Still, the finding matters beyond reproduction. The ovaries produce hormones that influence bone, cardiovascular health, metabolism, the brain, skin, and other tissues. Earlier loss of ovarian function is associated with broader long-term health consequences. Preserving ovarian function could therefore have benefits that extend beyond the possibility of pregnancy. Future research may examine whether reducing ovarian fibrosis can support healthier hormone production or delay some consequences of premature ovarian insufficiency. The study also changes how we visualize aging. We often imagine age-related decline as damage occurring only inside individual cells. DNA breaks, mitochondria weaken, proteins become disorganized. But cells do not live alone. They live inside a physical environment. That environment can become inflamed, crowded with scar-like material, and mechanically hostile. A healthy cell placed inside unhealthy tissue may no longer be able to perform normally. In the ovary, aging may partly resemble a garden whose seeds remain present but whose soil has gradually hardened. The researchers did not replace the seeds. They changed the soil. That does not solve the entire problem of reproductive aging. It does reveal a new place where scientists may be able to intervene. For women today, the practical message remains cautious. Age is still the strongest predictor of female fertility, and anyone concerned about ovarian reserve, irregular cycles, early menopause, chemotherapy, endometriosis, or polycystic ovary syndrome should discuss their individual situation with a qualified clinician. For researchers, however, the message is more provocative. The reproductive clock may not be controlled only by eggs and hormones. Part of it may be written into the stiffness of the organ itself. And if that stiffness can be safely reduced, one component of ovarian aging may be more reversible than scientists once assumed.